and please feel free to stop me. So, what I have in mind while creating this presentation is to provide a key functionality of allele registry that is important from the viewpoint of curator who will be using these allele registry identifiers in variant curation interface and gene curation interface. And I put up few slides in addition to this basic use case. So let me just go ahead now. And we all know that there are various ways a variation can be defined. And in various variant-centric resources as well as in literature, you will come across wide variety of representation of variation. And the data that is associated with that these variation are so much valuable that it can help to save life of patients. So it is very important part of overall our curation effort that we provide the variant identity completely, and it should at least be it should be dereferenceable and should be able to pulled out whenever some other ways the variation is described. So this is the ultimate objective of the ClinGen Allele Registry, and now I'll go in details of how it will be useful. So first question is, what are sequence variations and how they are defined? So basically, let me first highlight what is a reference sequence. And reference sequence is any sequence that you can use to refer some annotations on. So say for example, reference genome is a one consensus representation of genome made available by Genome Reference Consortium, and the sequence that they produce for each chromosome is called reference genome, and it has been revised many times, and there are three major assemblies available. Similarly, if there is what I mean by reference transcript and genomic sequence, reference genomic sequence. So these transcript and reference genomic sequence are accession and retrievable sequence of nucleotide or amino acid sequences that is present in some authoritative agencies like NCBI or EBI, and ideally these reference sequences are generally encouraged to have a stable identifier if they are supported by some evidence, experimental evidence. So that's what I mean by reference sequence. Any differences in this reference sequence is nothing but that's how you define the variation. So to provide a very basic example of what I mean by reference sequence is, say for example, I have this particular accession that is RF1, and it has a very simple sequence of 9 nucleotide ACT, GTC, GTG, and the position, if you go from 5' to 3', it is 1 to 9. And now I want to define a variation as a function of this reference sequence. So for that particular objective, what I need is first I need to put the reference sequence identifier. So I have put the reference sequence identifier. The second thing that I need is to, in this reference sequence, what is the position where I see my variation. So say for example, here is the position 4. So at position 4, I see a variation in this reference sequence. And what is the nature of this variation? Instead of G, I see it. In my sample, I see an A. So in order to define a variation, I need reference sequence identifier, position in the reference sequence, and what is the new allele that is introduced in this reference sequence. And this is how in a simple way to define a variation. And to come up with this representation of variation, Human Genome Variation Society has come up with very nice detailed documentation of how to define this particular information in form of one string. And it says it just simply says that you first provide your transcript, reference identifier, then you provide C dot if it is a transcript and G dot if it is a genomic. Then you provide position of the alteration. Reference is not absolutely required, but it is always good to provide reference in order to validate if you are talking about the reference sequence is at that position you actually see that nucleotide, and this is the alternate sequence. So this is a very basic nomenclature for substitution that is recommended by HGVS, Human Genome Variation Society. Now similarly, if you have variation in a genomic sequence, you can use the similar kind of identifier. But the major problem is there are multiple transcripts of one particular gene. Say for example, currently we have 41 transcript for TP53 and 16 for BRCA1, and we already have 3 genome assemblies. So basically I can define a variation as a function of any of these transcript or any of this genome assembly or any of the protein variation. So it becomes very difficult to understand if you are defining as a function of different transcript or genome assemblies. It becomes very difficult to collect the data for this particular variation. Now the other problem is insertion deletions. So there are different ways where these insertion deletions are described. Say for example, in the reference sequence that I showed in my previous slides, what I am doing is I am deleting 3 nucleotides from the sequence at position 4 to 6, and it gives me a HGVS expression that is RF1C dot 4 to 6 deletion. In the second variation, what I am doing is I am going at position 5, 6, and 7 and deleting those 3 nucleotides. In the third position, in the third variation, what I am doing is I am going from 6, 7, and 8. Nucleotide 6, 7, and 8 and deleting them. But if you notice very carefully, you will see that no matter if I delete 4 to 6, 5 to 7, or 6 to 8, I am going to get the same alternate sequence, that is ACTGTG, ACTGTG, similarly ACTGTG. So these are 3 ways you can define these deletions, and still they result in the same alternate sequence. And based upon the what kind of variant calling algorithm you are using, or what nomenclature you are familiar with, sometimes you get first type of expression, or you may get the last type of expression. And in literature I have seen the other cases also where there was no normalization has done, and it just shows some deletion in particular genome. And in addition to the 2 ways I described in my previous slide, there are plenty of literature you will find, and still in the reports, patient report, you will find that mutations are described like as a function of gene, similar to what I have shown here, and it becomes really difficult to unambiguously identify the variant that is described here. So how does the ClinGen registry help in such kind of cases? So if you have a variant identity, if you are able to identify a variant using HGVS expression, or if you have VCF files, or if you have VCF representation of allele, or if you have a partial information, you just know the gene and you want to define a variation, okay, so you can provide this identity to allele registry, and what it does, it produces this identifier of this sort, like first 2 letters are CA, and there is a numerical representation here, and what CA stands for is the canonical allele identifier, and that's the whole entire use case or function of the allele registry. And while going from the variant identity to providing the canonical allele identifier, it goes through several steps. First it makes sure that what you are talking is correct. It's not outside the reference sequences that you are defining. Then it maps to the main reference genome, and the current latest assembly is GRCH38, and it maps to that. It normalizes the variation on the fly, and checks if in the database, in the allele registry, if your variation is already registered. And it also produces it also provides a mapping to the various transcript, genome assemblies, and amino acid reference sequences. So, okay, so what is the use of all these, steps, and what is the use of this CI identifier, and I mean, how does it help to solve the problem? So this, if you follow this process, it the identifier, the CID that is produced, is become dereferenceable and retrievable irrespective of reference sequence and normalization status. So no matter how you define the variation as a function of genome assemblies or as a function of transcript, one transcript, another transcript, or you have a the three cases that I showed. If you search using the different types of HGVS expression which are non-normalized or normalized on the right or left, you are going to get the same identifier. So that's all about registry. No matter how you search variation, you are going to get the same identifier. So once it was ready, what we did is, to facilitate the job of curators, what we did is we went ahead and looked into some key resources like dbSNP, exec, nomad, myvariant.info, cosmic, and ClinVar, and we collected variants from these resources, and we provided identifier for each of them. So we have currently in allele registry 913 million allele identifiers from multiple sources. So it is very highly likely that the variant that you are looking for is already registered in the ClinGen Allele Registry. All you have to do is search that variant and get the identifier to use in VCI, GCI, or any other resource. So there can be two scenarios to get identifier. One thing is like you have a complete HGVS expression, and the other, in this scenario, what you need to do is you just go to reg.clinicalgenome.org website, and you will presented with this kind of page. And in the first search box, that is, I have enlarged it here. On the drop box menu, you have to select HGVS, and in the second box, you just write your HGVS expression. If the HGVS expression is valid, there might be two scenarios now. Once you search using HGVS expression, likely the variation is already in the ClinGen Allele Registry. If it is already present, then you are presented with this particular type of page, and on the top left you will see that this sort of identifier, and it is it clearly says that it is a canonical identifier, and you can use this identifier in curation resources. I can hear some background sound, Jenny. Okay, great. So this is my canonical identifier, and there are some additional information that is provided along with this identifier. Is the gene name on which your variant is mapped to, and we have several link outs to these key resources. As I said, when we import the variant from these resources, we store the cross-linking information, so we can provide a link out to individual resources here. On the top panel, I have HGVS expression. What we show is HGVS expressions as a function of genome assemblies, and on the bottom panel we show the HGVS expression as a function of transcript and corresponding amino acid changes. And if you notice that it has this two particular link out. The VV is for variant validator, and it has recently been published, and they say that it helps you to make sure that your variant is represented correctly. So feel free to use that, and make sure that the variant is represented correctly. We also have this main preferred transcript mark tag here, and what it means is that main is a consortium where they are coming up with one transcript per gene kind of approach, and it will be uniform across NCBI and EBI. So that particular representation is shown marked as a main preferred transcript. Please note that this main preferred transcript will not be present for all the genes. It will not variation in all the genes, because they only provided preferred transcript for 50% of genome. So for 50% of genes, you have this particular mark on the registry page. So basically, Yeah, to so summarize this, approach is like, first, if you have HGVS expression and if you search allele registry, you may end up in getting a ClinGen Allele Registry identifier. But if your variant is really unique, it is not present anywhere, or it is a kind of insertion deletion that has not been seen before, then instead of showing the CA ID, it will show a prompt of get identifier. All other information that I mentioned in the previous slide, like gene and mapping to the genome assemblies, mapping to the various transcripts, and providing amino acid changes, they will be present, but it only thing that will be missing is get identifier. So all you have to do is click on that get identifier, and if you are a registered user, you can provide your login and password, and it will provide you identifier instantly. So this is the scenario when you have HUS expression and your allele registry identifier is your variant has never seen in the we didn't see your variant before in the allele registry. So you can get instantly identifier, and that identifier becomes usable in variant curation and gene curation interfaces. But we all know that it is not that easy to get HUS expression just like that. So we enter into a scenario where we don't have a complete HGVS expression, but we have a partial information about variant. And what I mean by partial information about variant is we know that in which gene the variation occurs, we know the position, and we know the alternate alleles and reference alleles. So what we do is we have if you go to reg.clinicalgenome.org, you will see do not have transcript or HUS expression, and a button for gene and variation based query. So all you have to do is click on that particular query and enter the gene symbol in the first box, then select whether you are defining as a function of transcript or it's a protein based nomenclature, and then you provide position, reference, and alternate allele. So once you provide this information and click query, you are presented with a list of possible variation, and you can investigate each possible mapping and find out based upon the corresponding protein variations, or link out to various resources. You should be able to find out which particular variant that you are looking for. So this is one very useful service, and it is widely used. Just provide gene, C dot or P dot, position, reference, and alternate allele. You don't need to have any transcript information available with you. The another interactive service that we provide in such cases is first this particular button you have to select from the landing page is interactively generate variation. So once you click on that, you will be prompted to enter a gene symbol, HGNC approved gene symbol. So once you search for gene symbol, you have all the transcript listed, and then you want to generate a variation based on that particular transcript. You click on that, and we here show that we in the next panel we show the relative nomenclature of the numbering of the reference sequence. All you have to do is select and provide the alternate allele, and just click search allele. So without actually just the variation that the information that you are entering is gene symbol and selecting a variation here and search allele, you should be able to get a list of probable. You should be able to here you will be able to get the exact CID, because you are specifying transcript here and position as a function of transcript and alternate and reference allele. So you should be able to get the exact CI identifier. And now if you are a ClinGen Biocurator, I have just provided a workflow here. So how to go ahead with the as you know that if you are working on VCI and GCI, you will need to have a canonical allele identifier ClinVar variation ID to initiate curation in variant curation interface and to supplement case level data or other types of data in gene curation interface. So for this scenario, like first to initiate curation, variant curation, what you can initiate curation with ClinVar Variation ID, and ClinGen Allele Registry ID. So now, say for example, there are two possible ways you can get identifier. So if you are familiar with ClinGen Allele Registry, then you can directly go to ClinGen Allele Registry. But if you are familiar with ClinVar, and you can go directly to ClinVar and search your variation using partial information. If the variant is found, then you can use the ClinVar Variation ID in the variant curation interface and start curating the variant. It might happen that ClinVar does not have that variation identifier for that variation. Then you come to ClinGen Allele Registry. Then in ClinGen Allele Registry, you can search using HUS expression. You can search using the interactive ways I provided you in the previous slides. So you can search. If you found a variation and CA ID, then you can directly enter into the variant curation interface. It is likely, in some scenario it might happen that you don't have we don't have variation. ClinGen Allele Registry does not have identifier for that. All you have to do is just quickly get an identifier by clicking on get an identifier, and you can provide a ClinGen Allele Registry ID in the variant curation interface. So this is a general workflow if you are working with curation interfaces and you need the Allele Registry ID for the purpose of curation. But there are many more use cases that can be possible by ClinGen Allele Registry, just thus the ClinGen Allele as a ClinGen Allele Registry user. So as I said in my previous slide, that Allele Registry cross-link identifier from various resources. So you can search different variation using any of the identifier like ClinVar Variation ID or RCV ID or DVSNP ID, and all the information like it provides various ways to search Allele Registry. Say for example, you can search ClinVar using exec ID, or using exec ID you can search myvariant.info. So this cross-linking is very useful, and it extends the way how you can search different resources. And if you think that we all know that these resources are not exhaustive, and it is likely that you might want to add many more resources. So we have a way for that, and that's what I have shown in this slide. So on the left panel I have shown is like there are three key databases in the ClinGen Allele Registry. One is Allele database, where the CIDs are stored, the reference sequences and metadata database, where we store the information about various reference sequences and mapping, and third is the link database, where we store the link outs to various resources. And these are the various services or various predefined processes that goes in the ClinGen registry, that is validation, normalization, canonicalization, projection. And these links can be of two types. So one is core resources like ClinVar, Nomad, DBSNP, Cosmic, myvariant.info, Cosmic. So these are the resources that we maintain as a in the ClinGen Allele Registry. But we have a way to and if you are interested in one particular resource, you can cross-link by yourself. So one example of such resource is a Civic. We worked with the team and we provided them whatever information they need, and they were able to manage the linking by themselves. The another resources like a PMID and CID database that we have locally, where we mapping of various CID to PMID. And please note that this is not an exhaustive database. We want to keep adding the associations there, and it might happen that there are plenty of PubMed ID where they mention the variation, but it is not linked here because we just don't have that information. Similarly, if a CID lists 3 PubMed ID, it is not like that CID has only 3 PubMed ID. It might be that there might be many more articles describing that variant. The other resource that we have is allelic epigenome and evidence repo. That's a ClinGen database. And here I have put a blank box with a note that database of your interest. So if you know a valuable resource that you would like to see in Allele Registry, or you want to use in during your curation, then do get us in touch with them so we can provide them information, and they themselves will be able to manage cross-linking. The any resource will be able to manage their cross-linking in allele registry. And I would just like to highlight that the allele registry is a very valuable resource, and it has already been used by the identifier provided by allele registry has been used by various resources like ClinVar, EMBL, Civic, and of course, the first use case was ClinGen database of pathogenic variant myvariant.info and VICC consortium. So and so that's something good for that shows the usefulness of the allele registry identifiers. And as we know that every resource has some limitations, similarly allele registry has a limitations, and some of them can be addressed in future, and some of them some limitations may need future investigations and specialized methods to handle such cases. So the first limitation of allele registry, it does not provide identifier for uncertain breakpoints. And if your variation is more than 10,000 bases, then when it is projected to latest assembly, you cannot get identifier from the allele registry. These both limitations we are actively working on, and we hope that we should be able to provide identifier for both cases in next 4 months. And we need new methods, and the cases where we need few methods is new methods is like the current canonicalization model will fail if the substitution in genome affects the splicing or cause inclusion or exclusion of exon in the transcript. It is very difficult to canonicalize variants which has a different effect on genome and transcript. The another limitation is HUS expressions for an indel may be representation of small 2 or 3 variations, and there should be a way of fractionating these indels into smaller pieces, but currently we don't have that particular services. So these are the two key limitations where the canonicalization model fails. It is always good to keep it in mind that it's not perfect canonicalization. And in these slides, I have put up some key resources that might be helpful in understanding the allele registry in a much beyond this presentation. Is like we have published one article that was a part of ClinGen ClinVar special issue. You can go ahead and read it, or if you have the VICC consortium has also made the paper available recently on bioRxiv. It is a very good paper, and it shows the how the allele registry is useful in collecting information from the 6 cancer-centric resources. Here, in this article, we show the FDA recognition process of ClinGen database highlights. This document highlights the role of ClinGen Allele Registry provided identifier. There is an introductory video by Aaron Riggs that is provides more or less same introduction to what I have provided here. Then Andy presented on this call couple of calls back, and that presentation is available if you would like to go use it as a reference. And Alex, who is a PI, who is our supervisor, who he also has a presentation as a part of Knockout Mouse project presentation. And finally, in this slide, I have put a lot of people who contributed in the development and use cases of ClinGen Allele Registry. So with that, I'll be happy to take any questions if you have.
Great. Thank you so much for that, Ronak. That was fantastic. Does anyone have any questions?
Hi, this is Shruti. I have a question about the cross-linking. So if there is a variant that's not, that is in ClinVar, but not on the allele registry, and then we create an identifier in the allele registry, at that time, does it cross-link to the ClinVar ID, or does that have to happen manually later?
No, when we import the ClinVar, we cross-link along with the no, along with the import.
Okay,
But
so it
you
will
know,
happen
yeah,
subsequently.
yes, exactly. Yeah.
Okay, thank you.
So, Ronak, how often do you update the allele registry? So how often would you be importing from ClinVar or other sources?
So for ClinVar, we check for frequent updates. But for other resources, if you see that they are very stable resources and they themselves are not updated that frequently, so whenever there is a major release comes for this other resources, we update in allele registry.
Ronak, this is Deb. So I may have missed a part of this. I had to step away for a second. But how are you handling complex variants? Was that just asked?
So complex variation, you mean haplotypes?
No, I would say like larger deletions or things like that. So I mean, there's like periodically in trying to curate, you come across a variant in a paper. It's not in ClinVar, and then the allele registry doesn't seem to hold those examples either. Is that in development, or is that just
I see. So as I said, these are the current limitation,
in your future? Okay.
yeah, and we are addressing it actively, and we'll have that somewhere in the July or August we are planning to support this kind of variation.
Okay, and my next question is maybe a little bit VCI or GCI related, but when I use the CA ID instead of ClinVar, it's not rendered the same way in the VCI. They use some kind of, it's like this, they use the cDNA or the genomic variant when they show it, like in the list. And to me, that makes it incredibly hard to like go back and find the variant I'm interested in. Is that an issue on the VCI side or on the allele registry side? Does anyone else know what I'm talking about here?
Yeah, I do, Deb.
Yeah.
Yep.
Ronak, do you know what I'm saying?
Yeah, I didn't understand exactly what you're saying. Are you talking about the when you enter the CID and click on the start curation, there you see a list of HUS expression. Are you talking about that?
No, so unfortunately I'm on my iPad right now and I can't really pull it up, but it's
yeah, it's like when you pull it in from ClinVar, like if it's an amino acid change, for example, when you pull it in from ClinVar in the VCI or GCI, it will show the cDNA change and the P dot change as well, so you can immediately identify what the variant is. But if it's been pulled in from the allele registry, as Deb was saying, I think it just shows the G dot number. So you get G dot blah, 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 C to A, but you've no idea is that a splice site change or a missense change or whatever. It's just really hard to, immediately recognize that as
like everything in the screen now that you're showing is all probably from ClinVar because I can see the amino acid changes.
Yeah.
Okay. And this also carries over. Sorry, Stephen, and this also carries over to the evidence summary. So I think it would be really helpful to be able to have the codon and protein in that instance too.
So the issue is that and if a variant comes from ClinVar, then the VCI is able to know what the preferred name is because ClinVar computes a preferred name based on the transcript. If the variant only comes from the allele registry, there isn't this concept of what the preferred transcript is, and so that's why there isn't this option of what is the way to express it in the VCI outside of the genomic coordinate.
Okay, so it's something that's on the allele registry side.
So I think, like I think if as the main project is going on and as we're getting to preferred transcripts for each gene, being able to build that into the allele registry, which I think the allele registry does call out the main transcript, that having that then feed into the VCI so you can see it, I think would be incredibly helpful.
Yes, and just to add to that point, we have been working for that particular interaction with Stanford team, and we started with ClinVar, and we realized that in ClinVar some of the preferred for the same gene they use a different transcript for the preferred name. So I think that will not be feasible to provide. That might not be very comprehensible for the end user. So what we did is main provides just one transcript for genes. So and it has recently been released. So we have imported in ClinGen Allele Registry now. And as you might notice that some of the protein annotations are missing, that we are fixing in this coming issue. And after that, there is all the information that requires to generate this kind of preferred name for the use in variant curation interface. So hopefully, it will come in variant curation interface soon.
Great.
Yeah, sorry, I was going to add to that. I was just going to say that, so at the moment, like, if you're looking at a variant title, as Steve was saying, if it is in ClinVar, then we show the ClinVar preferred title for that variant. But as Stephen said, the issue is that if there isn't a designated transcript, because when it comes in from ClinVar, they've designated a transcript already, which is why they're able to generate that title with the variant in the change and everything. But so we currently, if we can get a single canonical transcript from Ensemble, because they show canonical transcript via an automated method as well, then we show that. And we construct a ClinVar type title based on that canonical transcript. But if there isn't an obvious canonical transcript, then we can't do that at the moment, which is why we then show that genomic representation, which we know isn't ideal. But once we have the main transcript data, which Ronak said only covers about 50% of protein coding genes at the moment, but once that's comprehensive and we have the full set, then instead of using that ensemble canonical transcript, then we'll be able to use that main transcript to generate as a similar ClinVar title for everything, for all protein coding genes. So what I would say is that basically once the main project is 100% up and running for all protein coding genes, then we'll be able to give you a better representation of the variants within the VCI.
Great. Thanks. So I have a question here too from Erica Smith saying that sometimes there's a variant in HGMD that she can't find in ClinVar. Is it possible to use HGMD entries as a starting point? I guess a starting point in the allele registry to search by the HGMD identifier?
No, currently it is not possible because I think, to the best of my knowledge, HGMD is a commercial resource and I'm not sure if somebody is in touch with HGMD, then if we can, collaborate with them and get the HGMD identifier. If we get that association of CID and HGMD identifier, then we can allow searching by HGMD identifier also.
Yeah, so right now it is just based on just figuring out the HGVS or whatever you can use to get the CAID basically from those papers.
And variants in HGMD, they should also be listing the genomic coordinate and the transcript and C dot. So those you could also go put into the allele registry.
Great, fantastic. Ronak, I had a quick question too about the interactive mechanism for searching the allele registry, which is really nice. So you were showing a transcript, and then you can basically make the change there, and it will figure out what the HGVS is. Can you do that for intronic changes too, or is it just on the coding sequence?
Yes. You can do it in the coding sequence only because the transcript itself just has the coding sequence, right?
Right. Okay, so there's no way to look at intronic. I was curious. I tried it before, and I thought that was the case. Sorry, go ahead.
You can search using these, fuzzy queries like gene and variation-based queries. There it is possible to search using the this position can include plus and minus. So there you can do that. Yeah.
Okay, that's good to know. And I had another quick question about PMIDs. I know you spoke about this during your presentation, and maybe I missed it, but for the PMIDs that are now available in the allele registry, can you just let us know where those are coming from? Are they, for example, if ClinVar submitters include PMIDs in their summaries, is that where most of those are coming from? Or are they coming from the interfaces? Where are these PMIDs coming from?
It is not from the interfaces. This PMID CID association is mostly from ClinVar.
Okay.
And tiny fraction might come from the to the de novo TB where they maintain the database of variation that is de novo. So they provide CAID and variation identifier and PubMed ID. So
Okay.
these are the two major sources, and yeah.
So
It is
that's good
not
to know.
comprehensive. Let me highlight, reiterate that it is not comprehensive. And if you have a list of variation associated with PubMed article, if you send it to us, or we can make sure that we include in that database, or we can pass control to you in terms of you can do it by yourself.
Okay, that's great. Fantastic. Yeah, that's it's a nice feature, and I guess over time there will be more and more in there, which is great.
Yeah, and again, the problem that we are having is we are limited by the association, right? So if you provide us association, we can make sure that it is part of this database. So even if you have spreadsheet of like variation and the PubMed ID, then we can make sure that it is included here.
Great, fantastic. Okay, so any other questions for Ronak? We have about 15 minutes left. No more questions. All right. Ronak, was there anything else you wanted to add before we leave the call? That was really great. Thanks.
Yeah, I don't want to add anything. Just, if you have any problems, feel free to reach out to us. And the address that I mentioned is in the last slide here, bcm.clingen@gmail.com. And we try to, respond to your request, considering we try to respond to your requests as soon as possible.
Great. Well, thank you so much. And it really is a terrific resource. So we really appreciate everyone's work on the Allele Registry. And